I'm Debra Silberg. I'm the Chief Scientific Officer, and I want to welcome everyone to the 2024 Beyond Celiac Research Summit. We are very excited about this. We've brought investigators from around the world who we've funded and some other academic colleagues to present their research. Beyond Celiac is dedicated, as a lot of you know, to finding a treatment and a cure. So the people that we've invited and the people that we're funding, that's their goal as well, is to get the data that we need to, and sometimes it's not the direct treatment, it might be the diagnostics or finding the ways that we can promote research. So we have here leaders in medicine, academia, our Beyond Celiac ambassadors represented from our board, and members of the celiac disease community. It's really an incredible mix of expertise and experience to draw on, and this is going to enrich our path towards a treatment and a cure. In the morning session, we will share, after the morning session, we'll share a link to participate in a session survey, and we'd be grateful for your response because then it can help us improve the Research Summit experience. So with that, I would like to start with the session entitled Predictors of Disease, Biomarkers, and Diagnosis. And our first speaker is Shalev Itzkovitz from the Weizmann Institute of Science in Israel. And his talk is entitled Analysis of Diagnosis of Celiac Disease via Shed Cell Transcriptomics.
Okay, thank you very much for the invitation to present. I'll tell you today about our Beyond Celiac funded project, Shed Cell Transcriptomics for the diagnosis of celiac disease. The gold standard for diagnosing celiac disease involves the acquisition of a biopsy through upper endoscopies. This is an invasive method, and while it's an amazing approach, it lacks molecular resolution. We basically histologically examine the tissue. We don't obtain molecular information. What I want to propose today is an alternative approach that can enable deep molecular profiling in a non-invasive manner of celiac disease using shed cell transcriptomics. Our gut has an amazing feature in that it sheds massive amounts of cells. Every day we shed about 30 grams of cells. A quarter of our feces is cells that are shed from the lining of the gut. The cells are born in these crypts, and then they migrate upwards along a few days along the villi axis as they absorb food and fight bacteria. And then when they reach the tips of the villi, they're shed. And the common thought is that once the cells reach the tips and dissociate, they immediately die. They dissociate from the tissue, they die, and that's it. We would only get, if we measure their content, we would only get the residual long-lived proteins. But a few years ago, we became very interested in mapping gene expression along the villus axis. We've done that in mice and recently also in the human villus. And we found that enterocytes constantly change their gene expression along the villus axis as they migrate. And when they reach the tips of the villi, they upregulate thousands of genes. And we asked, why would you want to invest so much energy in upregulating thousands of genes in cells that are going to die in a few hours? And so we became interested in challenging this, and in mice, we analyzed not only the tissues, but also the fecal content. We basically flushed the mouse intestine, and we isolated cells, and we found that the cells are viable. They have a nucleus. They have these bright dots, which are transcription sites and the RNA molecules. We even did the single-cell RNA sequencing of whole cells from the lumen of the intestine, and they were whole, they were diverse, and we calculated that they survived for quite a few hours. And that was very exciting because that means that we can examine fecal samples and maybe obtain molecular information on the cells that are lining our gut in a non-invasive manner. So to test this idea, we teamed up with the Shiva Medical Center in Israel and wanted to analyze inflammatory bowel diseases based on fecal washes in human patients. These are patients that are arriving for a diagnostic procedure of colonoscopy for inflammatory bowel disease, Crohn's and colitis. And we obtained both biopsies and also fecal washes. These are aspirated fecal samples that are obtained before the colonoscope goes in. And we transcriptionally profiled these samples using poly-A sequencing. So we enrich for the RNA molecules that encode proteins and using basically single-cell protocols. So we use unique molecular identifiers that enable very sensitive measurements. And to our surprise, we found that the fecal wash transcriptomics was much more powerful in diagnosing active inflammatory bowel disease compared to biopsy transcriptomics. Here in these plots, you can see this is a principal component analysis. Every dot is a sample. On the left, you see biopsies. On the right, you see fecal washes. Blue are patients in remission or controls. Red are patients with active inflammation, with relapse. And you can see that in the biopsies, it's not so easy to separate between them. And in the fecal washes, you see a clear separation. And the reason that this approach is so powerful is that fecal washes average cells that are shed throughout the gastrointestinal tract. So you're not sensitive to the precise sampling location of a biopsy. And also, they're highly enriched for the cells that are more relevant to the disease. When you're transcriptionally profiling a biopsy, you're obtaining muscle cells, deep stromal cells that are not directly relevant to these kind of diseases. In fecal washes, you get an enrichment of epithelial cells and immune cells that live on the boundary between the tissue and the lumen. Okay, so that was all fecal samples obtained during colonoscopies after 2 days of very massive cleansing of the gut from bacteria. And what we really wanted to do is to obtain similar rich molecular information from stool samples that can be acquired at home. And that poses a much more a higher challenge because 99% of the RNA molecules in stool samples originate not from the cells that are shed from the gut, but from bacteria. And so we devised this approach to get rid of the bacterial, to massively reduce the amount of bacterial transcripts in order to enrich for the human transcripts. What we do is we extract RNA from the stool, and then we mix the RNA with DNA probes that we designed to be complementary to very abundant bacterial RNA, basically the ribosomal RNA of the most abundant microbial species. And then we treat the sample with this enzyme called RNase H1, which is an enzyme that eats up DNA-RNA hybrid molecules. And so this way we get rid of the abundant bacterial RNA molecules and we enrich for the human RNA molecules. We apply our poly-A transcriptomics, and this increased signal massively. And we're very happy to see that we get a very rich molecular signal in IBD from stool samples. So here every column is a sample, a stool sample from either controls in green or inflammatory bowel disease in purple and red. This is Crohn's and colitis. Every row is a gene, and you can see that inflamed IBD patients have an upregulation of the whole module, including the classic genes like calprotectin, S100A8, 9, cytokines, etc. And just an important disclosure, I'm a co-founder of Trace Cells, which is a company that tries to commercialize this approach. Okay, so this was all the previous technology, and now we wanted to see whether we could somehow use these shed cell transcriptomics to diagnose and characterize celiac disease. And for this, we teamed up with our collaborators at the Schneider Medical Center, Professor Ra'anan Shamir and Professor Dor Shoval. And basically what we did is to take both biopsies, upper endoscopic washes, and also stool samples, and transcriptionally profile them. Biopsies and washes by poly-A sequencing, and stool samples by poly-A sequencing after this microbial depletion. So let's see what are the results. These are the results of the biopsies. Every dot here is a gene. The x-axis is the ratio of expression between celiac and controls, and the y-axis is significance. 55 celiac biopsies and 33 controls. And the red genes on the right are genes that are upregulated in celiac biopsies, and red genes on the left are downregulated. Upregulated genes include classic genes like interferon genes, antigen presentation. Downregulated genes include many of the absorption genes of enterocytes, apolipoproteins, iron metabolism proteins, transporters, etc. And then we move to stool samples. And here we actually were very, very pleasantly surprised to see massive gene expression changes between stool samples of celiac patients and controls. And we were surprised because we often sample this disease in the proximal small intestine, and these stool samples are obtained at home from the colon, from the rectum. And we didn't really imagine that we'll see so much signals, but the signals are very strong, and they include very logical genes. Again, antigen presentation genes like B2M, HLA, interferon genes are upregulated in celiac stools, and we have massive downregulation of apolipoproteins and other enterocyte genes. We also extended this study to stool samples from another center, the Soroka Medical Center, and we were happy to see that there is a correlation. Every dot here is a gene. The x-axis is the ratio of celiac and control stools in one center. The y-axis is in the second center, and there's a nice correlation, which is encouraging. And I think this is the most important slide in the talk for us. Here we compared the changes in gene expression between celiac stools on the y-axis and celiac biopsies on the x-axis. And for example, you can see on the upper right quadrant, every dot here is a gene. All of the red genes are interferon and antigen presentation genes that are massively upregulated both in the biopsies and in the stool samples of celiac patients. And on the bottom corner, you can see the classic enterocyte genes like apolipoproteins. They are downregulated in both stool samples and biopsies of celiac patients compared to controls. Here are some examples. Again, this is the villus axis. The blue gene is apolipoprotein A4. You can see that it's downregulated in both the celiac biopsies and in the stools, as are other classic enterocyte genes. And you can also see an increase in these interferon, antigen presentation, other immune-related genes, both in the biopsies of celiac patients on the left and in the stool samples on the right. And very interestingly, we see a significant decline in celiac stools of calprotectin RNA. This is, again, these two genes, S100A8, A9. These are classic markers of neutrophils and monocytes, massively upregulated in stool samples of inflammatory bowel disease patients, Crohn's disease, but they're actually significantly downregulated in the stool samples of celiac disease patients. So there's a differential signal here compared to Crohn. Other interesting measurements we noticed, these are transcripts that belong to goblet cells. There is an increase in goblet cell transcripts in celiac biopsies, but very interestingly, there's a decrease in goblet cell transcripts in celiac stool. And that could potentially indicate that goblet cells remain within the tissue for longer. They're shed less in celiac disease, potentially to facilitate a thicker mucosal protection. And there's an opposite trend in another secretory cell called Tuft cell, but I would not go deeply into this. Okay, so these are the main changes. And now the question is whether we can use these stool transcriptomics, these measurements of a few thousands of genes that are expressed in the lining of the gut, their measurement in stool samples to diagnose celiac and classify patients into celiac and controls. So this is a principal component analysis of all of the stool samples we obtained from celiac patients in black and from controls in green. And already here you can sort of see that there seems to be a nice separation. So if you give me a new stool sample and it hits the top green circle, I can say this is most likely a celiac patient, and if it is a control patient, and if it's the bottom black circle, it might be control. But of course, we want to build a classifier, and we've done that with several classifiers like support vector machines and others, where you basically separate the data into a training set and a test set, and you train your classifier, your boundaries on the training data, and then you test that on the left out samples. And it seems to work encouragingly well. Again, these are only 20-something stool samples of celiac, and we believe we can do better by building an even better classifier that's controlled for gender, age, and other properties. Okay, one last point is the washes. So I told you about the biopsies of celiac, and I told you about stool samples. And actually, this whole project started with endoscopic washes because we were sure that endoscopic washes, which are sampled from the same location where we sampled the biopsies, would be much more representative of celiac disease. And it actually turns out to be not the case. Here on the x-axis is the ratio of gene expression of genes in celiac and controls. On the y-axis is the ratio of gene expression in upper endoscopic washes. And there is no significant correlation, unlike stool samples and biopsies. And actually, there is sometimes an anti-correlation. So you can see on the left upper quadrant, enterocyte genes that are down in the tissue and actually up in the washes. And what we think is going on here is that these endoscopic washes capture fragments of cells, cells that are currently attacked by intraepithelial lymphocytes and are very fragile, and they do not make it all the way to the stool. Whereas the stool samples are enriched for cells that are normally shed from the tips of the remaining blunted villi. And so this is why we think actually the stool samples are more representative of the pathophysiological changes in the gut in celiac disease. Okay, so I'll summarize. I've shown you that stool shed cell transcriptomics can provide very rich molecular information and also biomarkers for diagnostics, non-invasive diagnostic of celiac disease. We can get information on turnover properties of cells. I have much more to say about that, but I will leave it to the break. By combining single cell atlases and shed cell transcriptomics, we can say which cell types remain within the tissue for a long time and which ones are shed more frequently. And again, this is because this is a non-invasive method and it's very rich, it could be an interesting approach to monitor patients after they start gluten-free diet and really assess in a non-invasive manner the biology of a large portion of the intestine that we never sample, right? We sampled the first 20 centimeters of the intestine. There's another 5 meters downstream. There's probably a lot going on there. And we think these two transcriptomics can provide the window into that information. So I want to thank our collaborators. First of all, I want to thank Beyond Celiac for the generous funding for this project. And our collaborators in Soroka Medical Center, Shiva Medical Center, and mainly in Schneider Medical Center. There's a very close collaboration with Ra'anan Shamir and Dor Shoval. And of course, my lab, mainly Rachel Freeman and Karen Bar-Halperin, who spearheaded this project. So thank you.
Thank you. We're going to have questions after the second speaker. So I would be remiss in saying that we want to also thank CHOP for hosting this whole symposium, and for our sponsors today, Takeda Pharmaceuticals, Sanofi, and Teva Pharmaceuticals. So I want to thank them as well. Our next speaker is Michael FitzPatrick from the University of Oxford in the UK, and the title of his talk is, Can we develop a robust biomarker to monitor celiac disease activity?
Thanks, Debs. It's a pleasure to be here. Thank you very much for the invitation, and thank you for the support of Beyond Celiac for the research that I'm going to present today. So the main focus of our research is whether we can develop a better biomarker for monitoring celiac disease activity, and that's one of the projects we've been working on in Oxford in the UK. If any of you have been to Oxford, this is the kind of gleaming spires in the centre, the historical buildings. Where you probably won't go on the tourist trail is the John Radcliffe Hospital, which is about 1.5 miles out of the centre. But that's where I work, up on the 5th floor, and that's where our clinical and research department are based. And I just wanted to start by acknowledging that this is very much a team effort, acknowledging the members of the lab who've worked on this project, and all of the collaborators, both scientific and clinicians in Oxford, who work on celiac disease, as well as our funders. So I want to start by, I guess, giving you my sell and why I think biomarker research is really important for celiac disease and for us to get the most out of potential new treatments for celiac disease down the road. And hopefully I'll pitch this talk at somewhere that those without a scientific background can understand as well. So the latest breakthrough that has reached the clinic in terms of celiac disease treatment was, of course, in 1953, when this chap, Willem Carel-Dicker, described the gluten-free diet in a really interesting experimental medicine paper, and he showed that he could improve these children with malabsorption by removing the protein fraction of wheat from their diet. But we now recognise that the gluten-free diet is far from an ideal treatment for celiac disease, and I'm sure many of you in the audience would agree. It can profoundly affect the quality of life of patients who are following a gluten-free diet. It is also an inequitable treatment, those with financial barriers or health literacy barriers to following the diet. And I think there's increasing evidence that actually, for some patients, it's not an efficacious treatment. People have ongoing symptoms and ongoing evidence of disease activity in their bowel. But because of the last 30 years of understanding the immunology of celiac disease, we live in exciting times where there are multiple potential therapeutics that are in drug development in celiac disease. Many of these are in phase 2 early clinical trials, but some of them will soon be entering the phase 3 registration trials that will potentially allow them to come to the clinic. And so potentially, in 10 years' time, I could have options to treat my patients with celiac disease in my clinic in Oxford. But what will I need in addition to those treatments? Well, we need to have clear and robust diagnostic criteria, but we also need good scoring systems, both of people's symptoms and also of the biopsies that we take to aid our pathologists in scoring them. But one thing that we really lack is a good marker of how active the celiac disease is in order to monitor, which doesn't involve repeated endoscopies. And what I think we need is what I describe as the calprotectin of celiac disease. So in inflammatory bowel disease, that's Crohn's disease and colitis, we have a non-invasive test which pretty accurately allows us to say how active somebody's inflammatory bowel disease is. We can use it to monitor their treatment, to see if they're responding well, or to see if we need to switch to a new therapy. And that was the sort of the premise from the BREAD study, our biomarker study. Can we develop a non-invasive biomarker? And there are two aspects of this in celiac disease. The first is, can we find a marker for the intestinal damage, the damage to the enterocytes, the epithelial cells that we just heard about, but also can we have a marker for the adaptive immune response, so the T cells that are involved in the celiac disease biology. And it may be that we need a marker for both of these, because sometimes these processes do not always go in parallel. And that led to the two aims of our study. First, using proteomics to study the different proteins in the gut in celiac disease, and then a study that we've got some sort of fresh data from, which is looking to see if we can see some of the T cell responses that we know are happening in the gut within blood samples. So the first part of this is using a technique called mass spectrometry to study all of the proteins that are in the gut in both health and celiac disease and see how they differ. And we use this technique called mass spec, which we take the proteins in a sample, we break them up into fragments, and we put them into this machine, which effectively weighs them and tells us what amino acids are in those proteins. And that can give us the identification of all the proteins in that sample, but also give us some idea of how much of each protein there is. And we've worked with Professor Roman Fisher in the Target Discovery Institute in Oxford on this. And to cut 18 months of research short into one slide, we've looked at 114 samples from 56 individuals with and without celiac disease. And these are samples, some of which are whole duodenal biopsies, so whole biopsies from the small bowel. Some of them are from sorted epithelial cells, so just the cells that line the gut lining, and then also serum samples from blood tests. And we've got some really high quality data in terms of the protein yield. So from our kind of latest experiments, we're identifying about 6,500 proteins from each duodenal biopsy and about 850 proteins from serum samples. So we're getting really high quality and very high resolution protein data from these patients. And I just want to briefly mention a couple of slides about our whole biopsy study. So this is with samples from 31 patients with and without celiac disease. And the first thing, as you can see on this PCA plot, the sort of scatter plot, is that the red and blue dots separate, so active celiac disease and healthy controls separate, and treated celiac disease seems to form a spectrum in between. So there are proteins here that seem to be responsive to the level of disease activity. And in this volcano plot on the right, we see a number of proteins that are downregulated in the biopsies in celiac disease. And these are, again, the same proteins that were mentioned in the gene expression study before. So the ones that are involved in the machinery of digestion, of breaking down the food and the macronutrients that we consume. And then a range of proteins that are upregulated in celiac disease. And we can look at those in other ways. We can look at the networks of proteins, so related proteins that are involved in similar processes. And we can see proteins that are involved in antigen presentation, in host defense, and in cellular proliferation, which are increased in celiac disease. So this proteomics study has technically gone very well. I think this is one of the most high-resolution proteomics data sets that is out there, and hopefully will be a great resource both for identifying potential targets for biomarkers, but also for the community more widely. Now, the second part of this project was looking to see if we can see if the immune changes that are happening in the gut can be detected at all in blood. And our previous research that's under review, as well as Arnold Hahn's really lovely work looking at the T cell changes within the gut in celiac disease, have shown that a number of the immune cells are activated and altered in celiac disease, particularly CD8 T cells or killer T cells. And we are interested in whether we can detect some of these immune cell changes that we know are happening in the gut within blood samples. And there is a certain logic to this. So if we give someone with celiac disease a gluten challenge, about 6 days later, we get a peak of immune T cells that are circulating in their blood that are activated and gut homing. So we know that sometimes these cells do enter the circulation, but can we detect them there? And this data is sort of hot off the press. In the last few weeks, we've got this sequencing experiment completed. So what we've used is samples from patients with both active and treated celiac disease. And we've taken paired samples from a blood test and from a duodenal small bowel biopsy. We've extracted out the CD8 T cells that we're interested in and then sorted particular subsets of those that we're interested in and done this technique called single-cell RNA sequencing, which is where we look at all of the genes that those individual cells are expressing. And we can also get information on what's called their T-cell receptor, which is the receptor which recognizes different proteins and antigens and is also a kind of a unique barcode for that particular CD8 T cell. And if two CD8 T cells have the same T cell receptor, we know that they're related to one another. So in this experiment, we've sorted 65,000 cells from these 4 patients from their blood and gut. And technically, well, we've got a few things cut. No, that's all right. We've got sort of high quality data with 41,000 cells with gene expression data and 31,000 cells with this T cell receptor data, which is particularly relevant to the question we want to ask. There are lots of different flavors of CD8 T cells in the gut. And what's nice is that in this data set, we can see those subsets. And in particular, a subset, a type of tissue-resident memory cell that we are interested in, which is highly prevalent in the gut in celiac disease. And the reason we're interested in this one is that these cells are highly activated, often proliferating, and are cytotoxic. So they look like they are kind of key players in the immunology of celiac disease. And what we can see in this data set is that in the active celiac samples, rather than the treated celiac samples, these cells look like they are actively proliferating and activated and are less so in the context of treated celiac disease. So it looks like they do alter when gluten is removed. And then the final thing, this is literally from this week, is to start to see whether we can see the same T-cell receptor in the blood and gut, these barcodes which tell us that 2 cells are related to each other. And happily we can, so the cells in purple on these plots are ones where we can identify a pair in the blood and gut within the same individual. So we can now ask the question of whether we can detect some of the cell changes that we know are happening in the gut within the bloodstream. So overall, I hope I've convinced you that biomarker research is a really important companion to the work that's going on to develop new treatments for celiac disease, that proteomics hopefully will lead to new biomarkers to monitor the activity of celiac disease without requiring repeated endoscopy, and that perhaps we can use some of these novel sequencing techniques to monitor the activity of the immune response in blood tests as well. So once again, thank you to everyone in our lab, in particular Sarah and Gundra, who were the research associates who led on the sample collection and wet lab work, and then Carolina, my PhD student, who is leading on the kind of complex analysis of this data. Thank you very much.
Okay, thank you very much to our speakers. We're going to have a Q&A. If you're interested in asking a question, you can come up to this microphone. I'd appreciate if you could say your name, and if you're affiliated with a hospital or organization or a university, if you could say what that is too. All these sessions are being recorded, so that would be helpful to the people who are going to be listening. I'm going to start with a question. Do you envision, and this is to both of you, do you envision that your research will enable us to forgo endoscopies in the future?
Yeah, it's a good question. I think I don't see our approach replacing endoscopies, at least for diagnosis. I think it could provide a way to monitor the disease over time. Yeah, so I think it would take more research to really establish the statistical power. Diagnosis through biopsy is very powerful. So, but of course, with all the risks, I think you don't want to do it frequently. And I think in that direction, this is something that these approaches can contribute.
Yeah, I would agree with that. I think in terms of diagnosis, maybe not so much, but in terms of monitoring, what would be great is you could have a test that you could go to your primary care provider or your gastroenterologist every time you're not feeling so well, or every 6 months or a year, and you could just check up and make sure that things are under control. And at the moment, we don't have a good tool for that, and nobody wants to have an endoscopy every 6 months to monitor things. So that would be the goal.
Claire?
Hi, I'm Claire Baker. I'm not affiliated with any research or university setting. I know, Dr. FitzPatrick, in your research you had active celiac disease, or people not on the gluten-free diet, people who had been diagnosed and I guess on the gluten-free diet in your samples. Dr. Itzkovitz, you were noting celiac disease patients, but I imagine they were active disease, like in the process of being diagnosed. Would you consider, or have you considered including diagnosed and treated celiac disease patients as an additional control to see what might be going on in there? What might be different as a result of having lived with celiac disease for some period of time?
Yeah, definitely. So all of our patients were patients at diagnosis before commencement of gluten-free diet. And definitely, since many of the signals we see are signals of enterocytes, and specifically enterocytes at specific locations along the villus axis, so that'll be very interesting. And this is a follow-up that we're planning to sample the acquire stool samples at diagnosis, and then every few weeks and months after commencement of gluten-free diet to monitor the mucosal healing process.
Thanks.
Hi, I'm Richard Flavel. I'm the founding chair of the immunology department at Yale, and I'm professor still working on celiac, among other things. So 2 great talks, by the way. Congratulations. I got one for each of you. So let's start here, since that's the easy question, and they're both easy questions, actually. But so it relates to T cells. So I know you were using 3' sequencing, which of course doesn't make it easy to get sequence from T cells. But I wonder whether you've done any sequencing so you could actually get the T cell receptor sequences, which are very, very helpful in understanding the disease from your stool material, which would be really fantastic.
Yeah, thank you for the question. This was surprising for us because we had quite a lot of experience with analyzing stool and fecal samples from Crohn's and ulcerative colitis, where most of the RNA signal we see originate from immune cells, macrophages, T cells. In celiac disease, very few molecules originated from the T cells. So I don't know why. Maybe it's segmental. There's more immune cells at the proximal small intestine, but the enterocyte changes carry on all the way to the ileum, or whether they're just more fragile in this disease in the lumen. So the majority of, most of the signal we get are actually from enterocytes. Unfortunately, not from T cells, but again, I think it's maybe combining this with proteomics, which are more stable entities, could also give an angle on the immune components.
Maybe the T cells aren't so stupid. They migrate away from that. I mean, there's no point from their point of view of getting sloughed into the gut.
Maybe.
Yeah.
Yeah, of course.
I mean,
of course,
they're migratory, right? So.
They're migratory, and they're more probably within the stroma. Although we do have these intraepithelial lymphocytes that are integrated.
Sure.
And we also don't see we get very low signal from them as well. So I imagine they might be a bit more fragile in the lumen compared to the enterocytes.
And on the also a beautiful talk on the T cells. So, what is the relative frequency of the antigen-specific cells in the gut versus the blood?
So these are the CD4 gluten-specific T cells. So,
well, yeah, or CD4 or 8s actually would be more useful, but you don't necessarily you know what's oligoclonal. Put it that way.
Yeah. In terms of the CD8 T cells, sorry, the CD4 T cells, the gluten-specific ones, in the gut, they perhaps only make up 5 to 10% of the CD4s that are in the gut biopsy. Yep. And in the blood, when they're detected by tetramer in Ludwig Solid's work, we're looking at like one T cell in 1 million to 1 T cell in 100,000 within the CD4s. They're extremely uncommon. In the CD8s, we don't know what any of them are responding to, if any of them are antigen restricted by gluten. There is no evidence for that to date. They are a very clonal population.
Exactly.
And I am sure that they are responding to something, whether that is a self-antigen,
Yes.
a microbial antigen, gluten via some pathway that's not been elucidated, we don't know. And in terms of how frequent they are in the circulation, I don't think we know. That's one of the reasons that we've asked these questions. We do know in the context of gluten challenge, indeed from Arnold Hahn's very nice paper in 2013, where you see after a gluten challenge this wave of CD103 positive activated T cells, which can make up 1 or 2% of the circulation in the context of a gluten challenge.
And so where you can see it, so this is quick, sorry,
Yeah.
where you
That's
can
okay.
see it, so the gut biopsy, how many clones are there of CD8s? Because the
Great
sequences
question.
that they get allow you to determine what the origins of the cells are and how many different codes.
Yeah.
I'm sure.
So we just, oh yeah, sure.
Thank you.
I don't know yet, but Arnold's paper from last year probably did show that.
They are certainly.
Say who you are first.
I'm Arnold Hahn. I'm the next speaker, actually. So we did describe that we think they're recognizing an antigen, as I will discuss in my talk. One issue is they're actually less clonal than like resident memory T cells, for example, or like effector memory T cells. So people have described, kind of paradoxically, when you take an active celiac disease patient and a non-celiac patient and do sequencing of their T cell repertoire, the repertoire of the active celiac patient actually gets less clonal, which is actually quite counterintuitive, right? But it's counterintuitive unless you consider that a very clonal population that already exists in your intestine is getting diluted by cells that are less clonal. Doesn't mean they're not recognizing something. Certainly, for instance, the CD4s are recognizing gluten, but it becomes a little difficult to discern that, yeah.
So you think that's a broadening of the response as the disease goes on to other antigens?
Yeah, I think that in a healthy intestine, there's high degree of clonality in cells. And this has been described going back, to spectra typing back, 30 years ago. So yeah, they're recognizing something, but it's not going to be so easy to pick out just by looking at clonality.
Yeah. But in your
Come back to the microphone because they won't be able to hear you.
but you have a number, right? Because you've
Yeah.
sequenced 27,000 cells or whatever it is.
Yes.
So how many clones of CD8s do you see?
So that plot we generated on Wednesday. So
Come on. It's already Saturday.
So you might have to give me a little bit of time. So that is one of
I'm kidding.
Genuinely, it is one of the reasons we did this.
Yeah, of
because
course.
We want to drill down on that question.
Right.
And
Yeah.
no one really knows, outside of the context of a gluten
Yeah.
challenge, whether you can detect those populations in the circulation
Yeah.
and at what frequency.
That's anyway,
So
very beautiful.
We'll find out.
Yeah. Thank you.
Thank you. I think we have 2 more questions.
Hi, I'm Amélie Terrien from Boston. I have a question about comparing like active celiac and tissue that is in healing, because I saw in your data that STAT3 was upregulated, but the JAK was like not compared to control. And STAT3 gain of function mutation have been implicated in like refractory enteropathies. And also in both of your data, like the CYP3A4, 2C9 were also downregulated, and it's involved in like drug metabolism. So do you have in your data the confirmation that those gene expressions are back to normal when the patient comes back into remission?
Yeah, in our case, not yet. We've only done that in diagnosis. And another critical, so we have a very good reference, spatial reference atlas for the healthy human small intestine, but we're very interested in understanding how that looks like in celiac disease, where the villi are blunted and then they regrow. So we know in the healthy small intestine, the tissue is beautiful. There's different layers that specialize in different processes. Now you have villus blunting, whether you have a situation where enterocytes now express multiple zonal programs within the same cell, or whether you completely lose the layers and you have sort of salt and pepper patterns. So it's sort of a complementary research that we're trying to push forward now, is to really describe what's going on in the tissue and in the shed cell fraction. But definitely, I think repeating these stool transcriptomics over time, that would be very interesting in order to see which of the normal programs come back and at what order.
So in the manuscript that we've got under submission, we included some treated celiac patients in our kind of immune epithelial single-cell sequencing atlas. And I kind of included them there as a kind of additional control, because I rather assumed that everything would have gone back to normal. These were samples a year down the line. People had responded well. Their biopsies were normal, according to our pathologists. And actually, they had lots of persistent changes, particularly in the epithelial compartments, that had not returned to normal, as well as in some of the tissue-resident memory immune cell populations. So some of those changes in the kind of absorbative and drug metabolism machinery had not returned to normal, even though the biopsies were reported as normal. Okay. And actually that's one of the main things our reviewers wanted, was more data about treated celiac disease, because that's, I think, one of the more interesting and surprising findings from that. Okay. And I think it adds to this idea that treated celiac disease may not be quite as well treated as we think it is.
All right. Thank you.
Thank you. And the last question?
Hello, thank you for the presentations. My name is Alan Ehrlich. I'm a family physician from Massachusetts and also the chair of the board of directors for Beyond Celiac. This is really for you, although, because you had talked about patients when gluten was reintroduced, seeing markers within like 7 days. You could tell differences. I think you said something about that. The reason I bring this up is one of the big challenges, particularly in primary care, is people come in who are already on a gluten-free diet. And then how do we diagnose them with celiac disease? They will adamantly refuse to do a 6-week gluten challenge.
I feel your pain.
Yes. Yeah. And so the question is, is there the potential in some of the work that you're doing, or perhaps with the shedded cells, to reduce the amount of time a gluten challenge would need to be implemented in order to get a signal that would convince us someone has celiac disease?
Yeah, I think that would be a great thing. So I normally put people on a gluten-containing diet for 6 or 8 weeks and then rebiopsy them, and that is just not practical for many people. It's really difficult. Honestly, I think the most interesting work from this has come from Jason Tydin and Bob Anderson's work in Australia, where they have shown that a cytokine called IL-2 increases in the circulation after a gluten challenge in people who have celiac disease and treated on a gluten-free diet. And I think they've published it. They certainly presented it at a conference last month. Really nice data that if you have people who are well treated, you give them a short gluten challenge, so potentially just 1 to 3 days, you get this, you can measure this really nice surge in this. It is hopefully on its way to being developed into some kind of commercial assay. But it would be great to be able to have a marker that you could do where no gluten challenge would be needed. And I guess that might be possible.
Do you know in that study you just quoted whether they used people with non-celiac gluten sensitivity as a control, or just non-celiac patients?
I think they did, but I don't want to tell you any lies, so I'm not certain.
Thank you.
So I want to thank our speakers very much. We have a 10-minute break, and we'll tell you when to come back. We have gluten-free snacks and water out in the lobby, so please enjoy.